Okay, guys. I think we'll get started in the interest of time. Thank you for joining us in the weekly epidemiology seminar. Today we have Dr. Nabiha Yusuf. She is a professor in dermatology in the Heersing School of Medicine. Her PhD is in immunology, and then she is also one of our own, as she has an MSPH from this school in epidemiology. She's an internationally recognized expert in skin immunology and environmental carcinogenesis. Her research focuses on innate immune responses, particularly the roles of TREM1 and KSRP in skin cancer development and immune suppression. She's had several federally funded research projects that I'm sure she'll tell us about today, and a Department of Defense grant. Her work bridges basic science and translational research, and she has a strong emphasis on health disparities in biomarker-driven epidemiological studies. She's a passionate mentor, and she's won multiple mentor awards from the graduate school and the MHERC. And she's published in really high-tier journals as well. And so I'm going to let her turn it over to her to talk about epidemiological insights into dermatological disorders. Thank you.
Thank you, Dr. Shrestha, for inviting me. And
I was supposed to say one thing.
is he online?
Do not forget to sign that paper in the front, okay? Thank you.
And thank you, Ryan, for the introduction. This is like this is not like a traditional epi talk. I'm going to talk a little bit about my journey, how I navigated between different careers, and how I landed in epidemiology, and how I went back to my lab, and then how I came back. So like it's like back and forth. So it'll give you like different flavors, and think about how you can flip between careers, and have so many options out there. So first few slides I'll talk about my journey, my academic journey, like my degrees and stuff like that. Then I'm going to talk about what is epidemiology of skin diseases, because we don't do that a lot here in UAB. So there's something to think about. Then I'll talk about some of our projects that we are doing, and some of the developing projects we plan to do, and I'm really looking forward to some good collaborations, and that is also one of the reasons I'm presenting here. Okay, so my journey. I'm originally from India, and I'm from like a state called Madhya Pradesh. Madhya Pradesh is the Hindi word. It's called like the central state. Madhya means central, like the central state. And I'm from a town called Jabalpur, which is like in the center of my state. You can see here, so we are like the heart of India. And these are some beautiful pictures of, we have this marble rocks where you can do like boating here, and this is a waterfall we have in my town. It's a very beautiful place. And this is my school where I went to, my high school. And then I did my bachelor's in biological sciences from this college. Then I did my master's in microbiology from this university. And this university also has an interesting story. It is named after a tribal queen. So my state has a lot of like forest and tribal areas, so it's very rich in that. Then I moved up north. I went to Aligarh Muslim University, which is a very renowned university in the north of India. And I joined a PhD program there, and this is like my medical college in Aligarh, where I did my PhD. So for my PhD, I actually characterized autoantibodies in dilated cardiomyopathy. So this was my PhD topic. It was all a human study that we did. And from there, you can see here, this was like Aligarh here. And then I moved up further north, and I landed in New Delhi. So in New Delhi, actually, I worked in New Delhi Tuberculosis Center as a bacteriologist. So this was my first instant at public health and what it means to take care of people's health. And basically, we were working on a project. It was a nationwide project where we were collecting samples from TB patients to develop a PCR-based assay at that time. And I learned something new there. This was like the DOTS program. I'm not sure how many of you are familiar with the DOTS program. It's like a short 6-month program. So they actually call the patient to the clinic to give them the medication. You know, multi-drug resistance is a big problem, right, in India and South Africa and a lot of other countries too. So the problem is patients do not take their medicines regularly and they throw it. So they call them to the clinic, and if the patient doesn't show up, they go to their homes. We have social workers who go to the homes and give them the medication. So that's why they were try to control the spread of disease. So this was my first stint at public health, you can say. And then after that, I moved to UAB. I came here as a postdoc fellow, the Department of Dermatology, Husing School of Medicine. And I've been there ever since. You can see I'm still in dermatology. And here I switched gears. Of course, I wanted to continue my cardiology research, but you know, when you apply from overseas, you try to get in, wherever you get in. And this was still immunology because I was doing immunodermatology. So we're looking at the role of immune system in skin cancer. I got promoted along the way, postdoc to instructor, assistant professor, and finally professor. And I got funded. My first award was the Career Development Award from the Dermatology Foundation. And while I was still in it was a 3-year award, while I was still in my second year, I got my 3 grants, 2 from the Department of Defense, 1 from the NIH and RO3. And I had to quit my award because you cannot be an extramural funding and a career development. So then I became assistant professor with that, and then associate and all that. And I also became a course director for 2 courses on campus on the GBS side. So I'm a course director for cancer immunology lecture course and the journal club that's offered like spring and fall, and the course is offered in spring. So while I was in my grants were ending, and I submitted my RO1, and I was like a naive first-time RO1 applicant. I didn't know where to submit. I submitted to a study section institute which did not have a new investigator benefit. So my grant kept hanging for a year, and I was not sure what will happen to me, my job and everything. So I started looking at some options, and I had like a postdoctoral fellow from the cancer prevention program in my lab that time, Erin Burns, and she was taking some epi courses as part of her requirement. And I actually took one class with her on cancer epidemiology, which was Dr. Waterboard's class. And you know how Dr. Waterboard is so interesting. And that kind of, gave me my plan. Okay, this is what I want to do. And I like to be among people, and I thought this was something like very natural, a good natural fit for me. So I took that class, and after I took that class, he became my advisor. He just suggested that I should do like a master's in epidemiology. Now, thinking about options, there's an MPH option and MSPH option, right? A lot of you are MPH students. So for me, the MSPH option was better because I'm a researcher. I write papers. I can write a thesis. So I said, okay, let me go with the MSPH option. So at that time, they had like a track which is not there anymore. It was like the pharmacoepidemiology and comparative effectiveness research. So I was on that track, and I took my classes, and it was a great experience. And one of my favorite classes was, of course, Epi 605, which was Dr. Jolly's Epidemiology of Infectious Diseases class. And because with my background in microbiology, I found it a little bit easier to do that. And while I was doing this course, I had to decide my thesis topic, right? So one of the requirements is you have to have a publication if you do an MSPH. So looking at different options, we had a pharmacoepi class, and Dr. Ryan Irwin, she was teaching the last lecture, and I was still deciding which area to go in, what my thesis topic and all that. And she suggested, she taught us a class on pharmacogenetics and pharmacogenomics. And I thought, okay, this is something interesting, more related to like the biology and stuff, So I said, okay, let me go with this. And she suggested I work with Dr. Stella Ashley-Bacon to do my thesis. So I have two members for my committee here, Dr. Bertha Hidalgo and Dr. Irwin. They were on my committee. And I decided to do a thesis with this golden study. It's the genetics of lipid-lowering drugs and diet network. So we did an epigenome-wide association study of inflammatory response to fenofibrate, which is a lipid-lowering drug. And this was like a secondary analysis. It was short, like a 3-week response of fenofibrate. And what we found that there were a total of, I'm not sure how many of you are familiar with DNA methylation, and I'm sure some of you are, I guess. So.
I was just told that your slides are not changing on Zoom.
Oh.
So I guess we are on Zoom. I don't know if you are able to spell easily, if you can share to Zoom. Apparently you're not sharing to Zoom.
Oh, okay. How do I do that?
Okay, I'm not.
Oh, yeah, okay. I'll just do the share thing, right?
Yeah,
It says I'm screen sharing.
even on Zoom.
Yeah.
If we can't get it, we can't get it.
Yeah, see, it says like screen sharing. Yeah.
Oh, she said it's stopped. It's working now. We're good.
Okay.
Whenever you're getting it. Cool. Thank you.
Okay, so we found a total of 7 CPG sites that were found to be significantly associated with this CRP, C-reactive protein, and 1 with IL-2 receptor and 4 with IL-6. The strongest association was found for the CPG site with the CRP IL-2 receptor, and this was located in this Kaya 1324L. It was on chromosome 7, and 7 of these top 10 genes were shown to interact with Kaya 1324L, and they were related to inflammation. So this was my thesis, published in Pharmacogenomics. If you want more details, you can look at this publication. Okay. So little bit of background about epidemiology of skin diseases, and this is like something new, and I thought I should touch a little bit upon this. So epidemiology of skin diseases, basically the distribution and determinants of skin diseases in human populations, involving research into incidence, prevalence, and risk factors of skin conditions. It's a crucial field for understanding how factors like age, ethnicity, and socioeconomic status influence skin health, and for developing prevention and treatment strategies. And skin diseases affect over 4.8 billion people globally and over 85 million in the United States. And they pose significant public health challenges because of high prevalence, cost, and disability impact. So this is a regional distribution. This is like a global distribution of skin and subcutaneous disease burden. So in a lot of African countries and in low-income countries, you see a lot of infection-related skin diseases, right? But if you see high-income countries like Australasia, and then you have high-income North America, you have more like melanomas, like cancers. So it just depends, and this is the distribution of all these diseases across the globe. Now this is by age, how these diseases are distributed by age. So you can see in the younger children, you have these cellulitis, pyoderma, and as they grow old, the pattern of diseases changes, and you can see in the higher age groups, you have more cancers, right? Because the immune system gets suppressed and all that. So this is a gender-specific prevalence of each skin disease between 2014 to 2018. So you can see the green bars are the males and the red bars are the females. So you can see the prevalence of each skin disease here. from atopic dermatitis, pigmentation, acne, and aging, rosacea, psoriasis, and all these diseases here. And this is simply the distribution of patients' time in the system, how much time they spent in the healthcare system. And this map actually shows you the age distribution of different skin or skin-related diseases between 2014 to 2018. So from these plots, you can see like diseases like atopic dermatitis, acne, and HS. This is called hidradenitis suppurativa. You can see it's more in the younger population compared to the older population. Now this is the prevalence of any skin disease. So you can see the prevalence is like 26 point, almost 27%, and this is the distribution age-wise. So you can see as people grow older, they have more prevalence of skin disease, and this is based on insurance status. This is like commercial population, and you see this is more in the 18 to 44-year-old category. This is the US Medicare population, where you have more skin diseases in the 65+ category. And this is the Medicaid population, which is more in the 0 to 17 category. And this is like the uninsured population, and you get that a lot in the 18 to 44 category. So this is the impact of skin disease on US life expectancy. This is the data from 2013, and I'm sure it's changed a lot after that. So there is something called YPLL, which is like the years of potential life lost. So you may have a projected life of say 75 years, but because of that disease, you died like at 50 years. So you lost those 25 years of your life, right? So this is like, I mean, I myself learned this new term, actually, YPLL, it's interesting. It's the years of potential life loss. If you had lived without the disease, you would have lived up to like 75 years. So this graph, this pie chart gives you like all these YPLLs for melanoma. You all know melanoma is very fatal, and then you non-melanoma skin cancer. And then you have all these different diseases, wounds, connective tissue, but mostly like the cancers. So the implications of this has importance in early detection and affordable treatment access. We need targeted interventions in high burden regions and integration of dermatological care into primary healthcare systems. We need public education and preventive strategies. That's what the job of public health is. Now I'm going to talk about my projects. We work on primarily on skin cancers and inflammatory skin diseases. So a little bit about skin cancer. So you all know UV is a major risk factor for skin cancer. This is UVA, UVB, and UVC, and they can penetrate skin at different levels. UVC is mostly cut off by the ozone layer in the atmosphere, but due to pollution, you get like holes in the atmosphere, and then you can get the UVC also, which is very dangerous. So we have different types of skin cancers. This is basal cell carcinoma, squamous cell carcinoma, Merkel cell. They are like non-melanoma cancer, and these are different types of melanomas: nodular, superficial spreading, and this is lentigo maligna. And these are the risk factors for skin cancer: high altitude with a sunny climate, moles on your skin, weakened immune system, exposure to radiation, poor diet, unhealthy lifestyle, photosensitivity, high arsenic exposure, history of precancerous lesions, fair skin, genetics, your family history of skin cancer, history of sunburns, then excessive tanning or indoor tanning, and then red or blonde hair, blue and green eyes. This is also a risk factor for skin cancer. So this is like a Fitzpatrick skin scale we use to measure, like you classify the skin type and color. So the first type is the skin color, which is a fair skin that burns very easily. and then this is the 2, 3. So these are the 3 categories that are most susceptible to skin cancer. Then you have other skin types 4, 5, and 6. So how does UV cause skin cancer? So excess UV, what it does, it forms pyrimidine dimers. Let me explain. Sorry. So it causes DNA damage. So it damages the DNA and forms dimers. So dimers can be direct damage or indirect damage in the form of reactive oxygen species. And these cause mutations in the tumor suppressor genes like P53. It can upregulate oncogenes. And then there's immune suppression, and UV causes immune suppression, and all these lead to skin cancer. There's like decreased apoptosis of the damaged cells, and they proliferate and cause cancers. They all lead to cutaneous malignancy. Now, in these couple of data I'm going to show you, we did like studies on single nucleotide polymorphisms. So what is a SNP? It's a genomic variant at a single base position in the DNA. So you can see here, if more than 1% of the population does not carry the same nucleotide at a specific position in the DNA sequence, then this can be classified as SNP. And they can be associated with certain diseases. They're associated with certain diseases, and they can have like a favorable outcome or a worse outcome depending on the role of that SNP. And this is very important because you can actually predict an individual's genetic predisposition to a disease, and they are also very useful in determining, like if you give pharmacological interventions to somebody and somebody has a SNP. For example, there was a paper in pharmacogenomics where they used imiquimod. So imiquimod is usually given for basal cell carcinoma, and it is also an agonist for toll-like receptor 7. So they found that people who have a SNP in TLR7, they do not respond to this treatment of imiquimod, and they had a very robust response. In like very few patients they were able to find an effect. So you can see how these SNPs can also predict drug response. So we did a study. This was Erin. This was my trainee from the cancer prevention program. And then her thesis was to look at association of vitamin D receptor polymorphisms with risk of non-melanoma skin cancer. So when we talk about vitamin D, we all know about vitamin D and UV, right? All that. So vitamin D, this vitamin D, it binds to the receptor called vitamin D receptor, which kind of binds with the retinoid receptor here, and they recruit co-activators. With the help of co-activators, they recruit RNA polymerase, and then of course this is a VDR response element, and they synthesize the protein. So there are certain SNPs present in VDR, so it's a vitamin D receptor. So these are the main ones that are reported, FOC1, and this is like the BSM1, TAC1, APA1. So most of the studies on vitamin D receptor polymorphisms were done in North America or Northern Europe. So there was no study in this part of the world, especially in our region. So we did this study. It was a small study. We only had like 200 participants. Like it's a case control study, so 100 controls almost and 100 cases. And we found particularly this SNP BSM1. This was people who had this were twice as likely to develop non-melanoma skin cancer. But then Aaron left, this project ended, we could not do. I wanted to study this further, but then, if you have other options, you just start getting into other areas, and we just left it at this stage. So now I'll tell you about my journey with TLR4, which I've been studying for more than a decade now. So this is a signaling cascade for toll-like receptor 4. So toll-like receptor 4 is a member of the toll-like receptor family. So for people who are not familiar with toll-like receptors or immunity, I can tell you something very simple. You have all travel on roads and you see a toll there, and you know what the toll does? You have to stop there, make some payment, there's a check, and then you go further, right? So they have the same function. So any infection or like pathogen associated pattern, these PAMs are pathogens, and the DAMs are damage associated patterns. So it's not, initially it was discovered to recognize pathogens or infectious organisms, but then people found that even damage associated patterns actually signal with this. So it sends a signal. When it senses these are patterns, they are innate immune receptors. So this is like they just sample this, and then they have this activate this signaling pathway that can lead to NF kappa B activation and late with the type 1 interferons. So we started this study. This was a part of my career development award. So we found that when we use mouse models to study. So when we have to study a molecule, we take a mouse which does not have that molecule. It's a gene knockout. So that gene is absent in the mouse, right? So this is a little schematic to show you how we studied this. So these are our mice. We gave them UV for we give them UV for 4 days, and then we apply this chemical hapten is DNFB. It's an irritant. So when you apply on the mice scratches, like massive scratching there. So you get a hypersensitivity response. It's called a delayed hypersensitivity response. And then after that, we apply it on the ear. So when you have this on the back, you have memory cells that develop. And then we have the second response on the ear. These cells migrate to the ear, and there's the ear swelling. So we measure ear swelling as an indicator. It's like an outcome of our response. So typically what happens when you give UV to the mice, before you apply that irritant, the response is suppressed because UV suppresses the response. And if you apply the hapten without UV, you have like a robust response. So you can see here from this that this is with DNFB, just the chemical here. You see these ear sections? You have like the massive inflammatory response here. Let me go back. And we did it in the knockout mice and the wild type mice. So the wild type mice have TLR4, right? These are the mice with the TLR4. And these are the mice without the TLR4, the top three. So you can see when you have TLR4, you get a suppression in the response, right? This is the positive control, you get a suppression. But if you see this with the TLR4 knockout, there's no suppression. So we think that TLR4, when you knock off TLR4, you are able to rescue the suppressive response. So this work was done by Tehseen, Wesley, and Eva. Eva got a medical student grant from American Skin Association with us. So then we did another experiment. We wanted to look at, immune suppression is a major factor for development of skin cancer. So this is what we study in our lab. So we wanted to see first whether DNA, it has any effect on DNA damage. So DNA damage, as I mentioned earlier, we measure, it causes like the cyclobutane pyrimidine dimers or CPDs in the skin. So you can see this green staining with the CPDs. And this is the wild type mice which have the TLR4. So we give UV to the mice and we harvest the skin at different time points. And these are the mice without the TLR4. So you can see there's a diminished response for the CPDs, the dimers that are formed. So this was done by my postdoc Israr, and Eva was also part of the study. Then we went on to do a carcinogenesis experiment in these mice. So these mice were on a black strain, so they were very resistant. So we used two mice. This another strain of mice which is like brownish. It's called the agouti strain, and the C3H-HEJ has a point mutation in TLR4. So we use these mice to study carcinogenesis, and these experiments were done by Israr and Tehseen. And you can see here, when you do not have TLR4, you can see like fewer tumors here. Then we got funded for another study to look at the TLR4 antagonist, how you can do this pharmacologically, how you can inhibit TLR4, right? So we used that inhibitor, and this study was done by Asif and Emma, then Min Chang. He was also one of our medical students here. And in this, we had 4 groups, 2 with UV, 2 without UV, and like with the vehicle and the drug, the compound TAC242 is called TAC242. So you can see here, like when you apply the compound, you had like fewer tumors here compared to the vehicle group. So I was always interested to see how we can study this in a human context, right? Because all this is all mouse work. So that's what I'm trying. The reason I'm showing you the mouse data in Epi seminar is to see how you can, translate this work to humans. So I've been doing this study before these mouse studies. We were looking at TLR4 SNPs in patients, and I was like, okay, I wish somebody had like a mouse which had this SNP. We were always on the lookout, and finally I did find a mouse which had the human TLR4 SNP. So this is what it is. So before that, we published an article, long due article for a clinical study that we did on TLR4 SNP and skin cancer. This was done by Dr. Sharfuddin and Outcomes Research, and this is Salim. So which SNP did we study? So gosh, this got a little bit messed up here. Okay, anyway. So this is a very common SNP what people study is D299G and then T399I. So D is the original amino acid, aspartic acid at this position. The position is 299, and this is the new amino acid that replaces G, right? So it replaces D. It's called glycine. And the other SNP is the threonine and replaces by isoleucine. So these two co-segregate, meaning they tend to be inherited together as a haplotype, and they're located in the extracellular part of the TLR4. You saw the TLR4 picture; it had an extracellular region and an intracellular. So this actually affects the extracellular region. So in this study, this is like the demographic clinical characteristics of our cases and controls. So there was difference between the age at the time of consent, and this is also a case control cross-sectional study, and we found some statistical difference in sunscreen reapplication. People who reapplied sunscreen had better outcomes, and people with actinic keratosis. Actinic keratosis are pre-malignant skin lesions. They regress on their own, or sometimes they develop skin cancer. So there was a significant difference here, so that's expected. Then we looked at the different SNPs here. This is like the two together. This is single D299G and T399I and K354K. And what we found that there was we did not find a significant difference, a p-value, but we saw a trend here. Like we found a p of 0.065 for association with skin cancer. So this is a mouse model I talked to you about, and this is what they did. They have a mouse homologue of 298G and 397I. So they are trying to superimpose here and tell us that, okay, this is very homologous to the human one. And it is like a knock-in mouse where you introduce a human SNP into a mouse. So we did an experiment. Now we went back and did an experiment to look at our UV-induced immune suppression model, like I mentioned before. This was done by Asif. And this is typically what you see when you give UV to the mice. That if you give UV, like in normal mice, they're suppressed when they don't respond to the hapten. But in the SNP mouse, you see there is less suppression here. So you can see the percentages here. On day 1 is 74% suppression in the wild type mice, is 45% in the SNP mouse. And day 2 also you can see these differences. So coming back to this little bit, so what we are doing now, we did study SNP in that human population, we published that, but we didn't know what's the functional significance of this, right? So we needed a mouse model. Now we have the mouse model. So what we are proposing now, we are working on a grant application. We are looking at the functional consequences of this SNP in mice, in immune suppression, in carcinogenesis. So we're going to use all state-of-art methods to see the, what is affected by if people have this SNP. So this is our ongoing study. So we are also interested to look at another, this is an inflammatory skin disease called hidradenitis suppurativa. It affects mostly people in the age of 20 to 30 years. It disproportionately affects women more than men, 3:1. And 1% of Americans have this disease. And 30% of those patients are African Americans or biracial. And it takes almost like 10 years to get a diagnosis. So these are different stages of the disease. Stage 1, you can have single or multiple abscess formation without sinus tracts or cicatrization. Stage 2 is recurrent single or multiple abscesses, widely separated and limited sinus tracts and this. And the stage 3 is diffuse or near diffuse involvement. So you can see these different pictures for the disease. This is the history, the disease course or natural history of disease. So you have a inflammatory phase, which is like reversible, this has basically inflammatory lesions such as nodules or abscesses. And if you have early diagnosis, you can actually prevent the disease at this stage, cure the disease. Then the destructive phase, where you have the both reversible inflammatory lesions and irreversible lesions, such as single or interconnected tunnels here. And then you have the burnout phase. So this is all the irreversible skin destruction you can see here. These are like tunnels, hypertrophic scars, fibrotic bands, indurated plaques, and contractures here. And this is the therapy. Drug therapy works like this, and surgical therapy is needed for these phases. And you have the adjuvant therapy, which can work for any phases, and of course lifestyle modification. So interestingly, like with my background back in cardiology, I was always trying to find some connection between skin and heart, or any kind of comorbidity, because I knew like this is like a systemic disease, and I'm sure there was a lot of studies done with psoriasis and cardiovascular diseases. And of course, this is also one disease which has a cardiovascular comorbidity. So it's just not cardiovascular. I can share this, show you this picture that it is associated with metabolic syndrome, which of course included all the cardiovascular things here, like arteriosclerosis, myocardial infarction, and then non-alcoholic fatty liver disease, type 2 diabetes, depression, sexual dysfunction, inflammatory arthritis, and inflammatory bowel disease. So all these these are like comorbidities associated with HS. So we try to develop like a cardiometabolic risk score for this disease in collaboration with Dr. Carrie Howell. And we had like we were just worked on the I2B2 data set, which is the UAB data set. And this was done by Charlotte and Salim. And as you can see here, the problem with the I2B2 data set is very small. And a lot of these values are missing for what we are looking for, all the labs for these patients. You can have a diagnosis, but you don't have the labs. Sometimes, it's like very hard to come up with really meaningful data. So what we just found with this right now is we found that these patients were obese. You can see 62% were obese. And they had, some of them had diabetes. And this is the BMI distribution. You can see there's a higher BMI associated with HS. And we also looked at insurance status, race, and area deprivation index. So you can see people with there are people with private insurance, and these were uninsured people, and these will have like public insurance, and there's African Americans were disproportionately affected, like I mentioned earlier, compared to whites. And then this area distribution index was all over, but they do have a higher ADI, and yeah, this is also like Victoria also contributed to the study, and she also got like a grant from the American Skin Association with us. And this is where we are right now. So we are looking at larger data sets because this is it's a big limiting factor for us. So that's where we are. We are looking at so we can derive some meaningful conclusions. And this is our kind of like where we are headed. So you all familiar with the social determinants of health. So they can be individual level or the neighborhood level, and they can be like race, marital status, insurance status. Neighborhood level can be socioeconomic factors, household, housing, rurality. So we think that they can be associated with severity of these diseases, and maybe that's leading to higher cardiometabolic disease burden and vice versa. So I would like to acknowledge a lot of people in my journey. So Dr. Elmetz, he was my mentor, my postdoc mentor, and we collaborated later on, and he was part of the studies, initial studies on TLR4, and of course the human TLR4 SNP study. That was his idea when I started it, when I was a postdoc actually with him. And Dr. Mazian, she was the director of medical genomics. Unfortunately, she just passed away last year. She was a big help with doing the analysis, genomic analysis for us. Dr. Waterbour, who actually got me into epidemiology, and he was part of the vitamin D study. Dr. Griffin analyzed the data for us, Dr. Russell Griffin, and Dr. Christina Hurst in genetic counseling. She was also a part, initial phase of the study. Dr. Sharfaddin, of course, she did the analysis for us, and Dr. Shu is my current collaborator, and he's the one I'm writing my grant with, the TLR4 SNP mouse model. And Dr. Howell, of course, for the cardiometabolic disease. My initial funding was from the Dermatology Foundation and NIAMS, NIAD, and we were funded by Erin was funded by the Cancer Prevention Program, and we got funding, 3 grants from the DOD, and 3 of my medical students got grants from the American Association, and 1 got from the Melanoma Research Foundation. And that is it, and thank you for listening. Excuse me if I went too fast.
I have a question. I assume there's a pretty active GWAS field are these traits? Are people working on like risk scores, like polygenic risk scores, or?
Yes, people have worked on polygenic risk scores, but not on like methylation risk scores. That's what something I'm interested in. We can do that. And Bartha and I also did like a health disparity education program, and we came up with a proposal in 23, right? 2023, yeah, where we looked at methylation and then income status and all that. So it's just the data is so hard to get. So we are looking at Trinetics and all of us other data sets. So yeah, but I'm really interested to collaborate with you guys. I know you have access to all the data sets, and suggestions are welcome. Yes.
I was wondering with HS, you said that it takes about 10 years to get diagnosed. What contributed to that? I imagine seeing a dermatologist probably.
Yes, so lot of things, yeah. So the thing is, like, initially, it's like you can say in a lay language, acne gone worse, you know. So first of all, access to dermatology clinics, then insurance status. If you don't have insurance, you go there. You don't go to a dermatologist, and you may not even think it's the disease. It's going to be so bad. The other thing is, like, even if you get the disease, they start you with on antibiotics initially because a lot of bacterial infestations there. So you think, okay, you know, but then when it gets worse, you need treatment with biologics, like, you know, like TNF inhibitors and IL-17 inhibitors have come on the market now. So it's the cost is high, right, for biologics. So a lot of people just stay on antibiotics, and it really doesn't help, you know, in the long run when the disease gets worse. So those are like a lot of barriers there. So we think a lot of this has to be associated with all this SES stuff. So this is what we are trying to, you know, dissect here. Yes.
What percentage of disease progresses in your heart?
I really don't know the number right now. Yeah.
But it's like 2% less than that, more than that.
Yeah, it depends like how soon you get diagnosed. If you get diagnosed at an early stage, probably you can take care of it. But you know, like it doesn't get diagnosed early, so if you go to a later stage, and yes, but
Since there's so many students here, one thing I really appreciated in your talk was how you find ways forward, like a water stream, sort of. Can you share with the group how you think about developing scientific questions? One thread I think was connecting skin to heart, for example, right? You also have a really diverse listing of funding, so you pursued a lot of different funding opportunities.
Mhm.
And you've sponsored, it looks like, a lot of students to also do research with your group. Could you share sort of how you think about science in that way?
yeah. So any research question you have. So first of all, try to dig in. You know, read. First thing is read a lot. Find good mentors. That's something. And mentoring is. I have a passion for mentoring. So like people just automatically, you know, come to me, and find something like out of the box. And usually it just comes from reading papers, finding grant opportunities. I think there are a lot of grants out there with this current scenario. I think we have to look for opportunities, you know, besides the regular mainstream grants. And there are several grants for medical students. I'm not too sure about public health, but I'm sure there are, like, you know, fellowships. And even with a background in public health, you can do some of the studies like biomarker related studies, and associate with a lab, you know, do part of your thesis like that. You know, like how I found my thesis very useful because I was able to integrate my background knowledge with, you know, my epi. So something like that. So talk to your advisor, talk to your mentor. I'll be happy to talk to anybody. I'm across the street in Walker Hall. So my doors are open. Anytime you can email me. I can just talk to you about any career trajectory you want to take or find any opportunity.